And welcome to our video lecture on topic 6.3, defense against infectious disease. Our essential understanding for today: the human body has structures and processes. We're going to talk a lot about those structures and processes that resist the continuous threat of invasion by pathogens. Lots of objectives. We're going to look at some vocab. We're going to talk about non-specific or innate immunity. We're going to outline the clotting cascade. This is what makes scabs. We're going to outline the activation of specific immunity, and then we're going to talk about how antibodies do stuff. We're going to describe the secondary immune response that we have after an initial infection or after a vaccination. We're going to describe production and uses of monoclonal antibodies, an amazing piece of technology. We're going to describe allergic responses really briefly. We'll outline how antibiotics work, how they treat some diseases but not others, and we're going to wrap up with a look at HIV and its effect on the immune system. First up, this guy Fleming, my spirit animal. If you've seen my desk, you know it's a little messy, but holy smokes, look at Fleming. He is, I aspire. I aspire to this kind of desk. So Fleming went on vacation one time, left his window open, and stuff blew in and fell on his petri dishes. And he came back from vacation. He was like, weird, some of my bacteria died. And then he discovered penicillin, which was secreted by fungal spores, fungi that started growing on his petri dishes with his bacteria. So these other two guys, Flory and Chain, they took over research in penicillin, and they were able to isolate it and use it as a treatment for diseases. So their first test, they picked 8 mice, made all of them sick. 4 of them got penicillin and they recovered, and the other 4 died. That was sad for those 4 mice. So Flory and Chain were like, awesome sauce. We have data from 8 whole mice. Let's go ahead and test on people, because, you know, 8 whole mice. So they tested on people, a man who was about to die from an infection, and they gave him some penicillin and he got better, but then they ran out of penicillin and so he died. So that was bad. But then they were like, we've got more penicillin. And 5 people who were suffering from infections got the penicillin and they all got better. And we were like, yay, amazing, Nobel Prize winning. And of course, if I'm asking you about ethics of something, there's probably some serious ethical issues, and definitely this would not be compliant with how we test new drugs today. And on that note, let's talk about some vocab. So pathogens. Pathogens are disease-causing agents. So anything that is going to cause an infection is a pathogen. Lots of different kinds of pathogens. Antigens are actually pieces of pathogens that trigger immune responses. So the antigen is actually what our immune system recognizes as being foreign, and the antigen is what we respond to. So we could have a pathogen that was super big like this. So here's maybe a bacteria cell, and here's one little protein on that bacteria cell. That's actually what our immune system will recognize, bind to, and then initiate an immune response. So the antigen is a little piece of the pathogen, and it's what our immune system actually recognizes and responds to. Leukocytes. Leukocytes are white blood cells. They are blood cells, and so they are part of our, and I always say this wrong, hematopoietic processes that lead to the differentiation of lots of different kinds of blood cells. So we talk a lot about erythrocytes. These are red blood cells. We're going to talk about these guys, macrophages. Macrophages, also called phagocytes, are one kind of white blood cell. These guys literally eat. Phage is eat. They're going to eat pathogens. They send out little extensions of their cell membrane and then gobble these guys up in vesicles. We also have lymphocytes, lymphocytes that are going to spend a little bit more time in the lymphatic system. We're going to talk mostly about T cells, B cells, and plasma cells, but clearly there are other kinds of lymphocytes in the paths to becoming immune system cells. Antibodies versus antibiotics. These sound almost the same. Notice how similar these words are, but they are very, very different things. You need to make sure that you keep them very, very clearly different. Antibodies are proteins that are made by these plasma cells. So plasma cells are antibodies. Antibodies are proteins made by plasma cells. Whoa, that was a struggle. Antibodies are proteins made by plasma cells. They are going to do all kinds of amazing things, binding to pathogens, preventing them from getting us sick. And we'll talk a lot about the functions of antibodies later. But for now, antibodies are proteins made by plasma cells in our bodies. This is part of the immune response. So we do this, we make antibodies as part of our own immune responses. Antibiotics, these are medicines, drugs that, like penicillin, often derived from fungi, that can kill bacteria. And so we can use these antibiotics, this medicine, to kill off bacteria, but we also have antibodies that our own bodies make that will help to kill the bacteria and viruses and all kinds of other things. So be super careful, antibody versus antibiotic, they look the same, they are not the same. So that word pathogen means a disease-causing agent, something that makes us sick. I want to know which kingdoms have pathogens in it. Definitely fungi, right? Athlete's foot is a fungus. It's a pathogen. It makes us sick. Protista. These are things like amoeba. If you've ever heard of amoebic dysentery, this is definitely a pathogen. Malaria is also a protist. So these are eukaryotes, single-celled eukaryotes. Animalia, worms, right? Tapeworms, hookworms, those parasites are definitely pathogens. They can make us sick. Eubacteria, all kinds of bacterial infections can make us sick. Archaebacteria, interestingly, don't make us as sick as do the eubacteria, just because they don't tend to live where we live. If they did, they might make us sick, but because they don't survive where we survive, it's kind of a non-issue. Plantae, I did some research, I could not find any plants that make us sick. So we have lots of like poison ivy that could give us an allergic response. Maybe you could call that a pathogen, kind of, sort of, but not really. So anyhow, definitely animals, fungi, protists, and eubacteria, not so much plants or archaebacteria, but pathogens kind of all over the place in terms of the wide kingdom of living things. So let's start talking about immune responses. We have structures and processes that resist the continuous threat of invasion by pathogens. We are constantly under attack, my friends. Right now, there are germs in your body trying to make you sick. Our primary line of defense is going to be the skin and then mucous membranes that are just inside any orifices, inside our nose, inside our mouths. We have in those down the trachea, down the nasal passages, we have mucus being secreted by these goblet cells. So these goblet cells in here secrete out some mucus, and that mucus kind of just is sticky. And so those germs, pathogens will stick to the mucus. We make lysozyme in that mucus that's going to kill off, break down those pathogens that get stuck in the mucus. Then we also have some cilia along the mucous membrane that is going to kind of like flush that mucus out. And so anytime you're like trying to get some phlegm out of your throat, it's because those cilia have been moving that mucus around. We want to get that mucus out because it's full of dead pathogens. So lots and lots of orifices, holes in our bodies have these mucous membranes, lysozymes, cilia to help prevent anything from getting into the inner sides of our bodies. Let's say that we have a cut in that skin. We don't want pathogens getting in. We're going to seal up those cuts with blood clotting. And so this is one of these processes that is going to resist infection. So we have this thing called the clotting cascade where we have a system of reactions where one thing triggers the next thing, triggers the next thing. And so this is a lovely waterfall of reactions. And the first part of the reaction is that these clotting factors are released by damaged tissue. So when the cells get broken, these tissues are damaged, clotting factors are released. We have also platelets floating around in our blood all the time. They're these little cell fragments in our blood plasma. When there is a cut, these platelets and clotting factors are going to activate the conversion of this protein from prothrombin into this other thing called thrombin. So we have this floating around all the time. The clotting factors that are released during an injury are going to turn that prothrombin into thrombin. That thrombin, this activated version of this molecule, is then going to change this thing called fibrinogen into fibrin. Fibrinogen is a soluble globular protein that floats around in the blood all the time. Fibrin is very fibrous and insoluble, and it is going to bond to lots of other fibrin molecules, and it makes this mesh of fibers across the wound, and blood cells get stuck in it, and that is a scab, which is amazing. So here's a picture of all those words that we just said. We're going to have these clotting factors and platelets that are released during any kind of tissue damage. Clotting factors are going to convert prothrombin into thrombin. So we're going to convert prothrombin into thrombin, clotting factors, and then also those platelets do that. Once we have this thrombin, thrombin is then going to change fibrinogen into fibrin. Fibrin is this mesh-like structure, and then those blood cells trying to get out of the body because there's more pressure here, less pressure here, so they're trying to flow to that area of lower pressure. They're going to get caught in these fibers, this fibrin mesh, and then we have this scab, and it is going to plug up that wound, and it is amazing. Another amazing thing that happens when we have any kind of injury or infection is the activation of macrophages. So macrophages, these giant white blood cells, macrophages also called phagocytes, what they're going to do is engulf pathogens. So we're going to call it non-specific or innate. So innate or non-specific immunity, because these macrophages will gobble up anything. So this picture is showing that we've got a splinter. The splinter has some pathogens on it. These phagocytes, macrophages, actually ooze out of the capillaries near that wound due to these guys releasing some chemical signals that trigger an inflammatory response. So when things are swollen and a little bit warm, it's because of these mast cells, but this is good because it's bringing more immune cells to this area, which is why it gets swollen, because you've got all these cells doing stuff. So the macrophages, those phagocytes, are going to gobble up these pathogens through phagocytosis, and then magical things happen next. So once we have the non-specific response from the macrophages, so the macrophages are going to gobble up the pathogen, they're going to digest it, and that digestion is going to lead to the production of some tiny little pieces that our immune system decides are antigens. And then the tiny little pieces of bacteria that got chopped up, this cell is going to present those pieces of the bacteria or whatever the pathogen is on its own cell membrane. We're now going to call this guy a dendritic cell. We can also call it an antigen presenting cell. Antigen presenting cell, because it is literally presenting the antigens to the rest of the immune system. So macrophage is going to present the antigen to first up our helper T cells. So we're going to activate our helper T cells when we bind the antigen that is on the dendritic cell, that macrophage that gobbled up the bacteria and presented the antigen. We're going to bind, notice that we're binding that helper T cell to the dendritic cell where that antigen is, and then we're going to activate that helper T cell. So step 1, macrophage gobbles up. Step 2, helper T cell is going to bind to the antigen on that macrophage that is presenting the antigen. We're going to activate those guys. They're going to go through lots of mitosis. We have so many of them. These guys are then going to activate another kind of immune cell called a B cell. So you have lots of B cells floating around in our blood. The helper T cells will activate those B cells. And now I have activated B cells. They're going to also bind to that pathogen. We're going to send these guys through crazy amounts of mitosis. We call this cloning. So this is clonal selection because we are selecting to make lots of copies of the same cell. And then these guys start pumping out oodles and oodles of antibodies. And that's the goal, is that we make lots of antibodies, these protein molecules that are very specific to a particular antigen. Those antibodies can then go do all kinds of work to fight that pathogen. So here's that same picture. It's super small on the previous slide. We're going to make it a little bit bigger here. Again, we've got innate or we call this non-specific immunity. Microbe gets into our body despite all our efforts of the skin and mucous membranes. Macrophage is going to endocytosis, gobble up that pathogen. It's going to chop it up into little pieces, and some of those pieces will be presented as antigens. And so this dendritic cell, the macrophage, literally turns into a dendritic cell when it becomes an antigen presenter. Antigen presenting cell. So that antigen presenting cell, the macrophage, with its antigen being presented, is going to bind to and activate a helper T cell. So we've got helper T cell here. Helper T cell is going to bind to the macrophage with the antigen. It's going to go through lots and lots of mitosis, so much mitosis. And now we have lots of cells, lots of these helper T cells that are all going to be on the search for this particular antigen. They're going to activate some B cells. So we've got lots of B cells that are not yet specific, but as soon as they are activated by a helper T cell, they become specific to this exact pathogen because of that antigen and that helper T cell. So now we have an activated B cell. That B cell is going to go through so much mitosis. One more time, lots of mitosis. And then I've got lots and lots of clones. Again, we sometimes call this clonal selection because we have lots of copies of this same kind of cell. And these plasma cell clones are going to pump out so many antibodies, these proteins that go and help us fight the disease. So here are some of those same words that I just said. Activation of those helper T cells when we bind to that protein on the plasma membrane of that antigen presenting cell, that macrophage. Once we have an activated helper T cell, we can activate the B cells. Good, so the helper T cell's job is to activate the B cells. Activated B cells will reproduce super rapidly via mitosis. We're going to call those the product of this clonal selection, this activation of the B cells. These are going to be plasma cells, and then they've got so much ER and so much mitochondria, and they're going to make lots and lots of these proteins called antibodies, which are then secreted from the cell via exocytosis. And then they go and they fight the infection. It's amazing. Here's an antibody. So antibodies, we can also call them immunoglobulins. They are proteins. Antibodies are proteins, immunoglobulins, that are produced by those plasma cells, those plasma B cells. They are specific to each antigen. So we don't really have antibodies that will fight just any old disease. We make antigens that are specific for each and every one of all the diseases that we have ever encountered in our whole entire lifetimes. You have antibodies specific to each and every one of them. Structure of an antibody looks like this. We have these things called heavy chains. They're longer, so we call it a heavy chain. And then we also have these lighter chains. The ends of the light chain and the heavy chain are variable regions. And so these pieces, the very edges of the Y-shaped proteins, are going to be specific to only one antigen, whereas the rest of it is a constant region. So all of this part is the same for all of our antibodies, but these parts are specific to all the different kinds of antigens that we have ever been exposed to ever. What do antibodies do? Why are they so amazing? They have so many jobs. They might bind to a pathogen with those antigen binding regions, and that's going to make it easier for macrophages to come and find those pathogens and gobble them up and eat them through endocytosis. They might prevent viruses from being able to dock to host cells. So viruses have to insert their nucleic acids into a host cell in order to reproduce and make us sick. If the virus cannot get into our cells because it's stuck to an antibody, then it can't make us sick. Some antibodies will neutralize toxins that are produced by pathogens. Sometimes antibodies will actually dig holes through cell walls of bacteria cells, and then the cells will rupture, they'll burst because of stuff getting in and out. And then also, and this is kind of related to this binding part, so we can have lots of antibodies binding lots of bacteria together, or viruses or whatever, and then this agglutination, sticking together of lots of pathogens, prevents them from entering host cells, also makes it way easier to for macrophages to go and just gobble them up all at once. Here are all of those functions courtesy of BioNinja in an amazing image, and they also bring with us a mnemonic device. So antibodies panic. So antibodies actions, remember panic, precipitation. This is where they are going to bind them all together, makes it easier for them to kind of settle out of the blood, not get moved through the bloodstream anymore. Agglutination, sticking them all together. We're going to have clumping. That clumping can then help with the white blood cell binding to and then engulfing, gobbling up those pathogens. We might have some neutralization, especially if we're talking about some toxins. Inflammation, we're going to trigger histamine release. Histamine is one of those chemical signals that stimulates inflammation. Inflammation is going to lead to more immune cells moving to the area where we have an infection. And then complement. Complement is that drilling of a hole through a cell wall or cell membrane so that the cell will then explode. Exploding bacteria cells cannot make us sick. So antibody actions, you can remember panic, but don't panic because you know panic. So we've talked a little bit already about B cells and T cells. Now I'm going to throw in another cell, memory cells. So B cells, and we call them B cells because they mature in the bone marrow. All of our blood cells start in the bone marrow. B cells stay there to finish their maturation. T cells actually move to the thymus. The thymus is kind of at the base of the throat, kind of where the collarbones come together there on top of the sternum. So thymus is where the T cells will complete their differentiation. They will mature there in the thymus. So we're going to have these helper T cells. We're going to activate our B cells, make these plasma cells. We have one more kind of B cell that is amazing called our memory B cells. So memory cells are memory B cells. Even after the infection is killed off, those memory B cells are going to be floating around in our blood all the time. Super small numbers, but they are there all the time. So think about all the things that you have ever been exposed to, all of the diseases that you have ever had. You have memory cells for almost all of them, assuming that you've been exposed to that disease more recently. So anyhow, these memory cells are going to roll around in your body for long amounts of time. And if you ever see that particular disease again, those memory B cells very rapidly are going to respond. They're going to go through lots of mitosis, make lots of themselves that are all going to make lots of antibodies. And you're going to kill off that infection before you even are actually infected. So we can kill off those pathogens before they have a chance to multiply and make us sick. So this lovely graph is showing us that if we are exposed to a pathogen here, it does take us a little while because we've got that macrophage needs to gobble it up. It needs to present the antigen. We need a helper T cell to be activated. The helper T cell needs to activate the B cells. The B cells need to go through clonal selection and differentiate into plasma cells. And then we finally have plasma cells starting to make some antibodies. And then we can have these antibodies that are going to kill off the disease. And then we don't need those antibodies anymore. And so the numbers go down because we don't need them. Let's say that I catch that same germ again. Maybe I was sick here and I coughed on my brother and now my brother is trying to give it back to me. Oh no, not going to happen, because what is going to happen is this. As soon as that pathogen is inside my body, those memory B cells are going to be like, wait, I remember what this looks like, and watch me make all of the antibodies, so many antibodies. Notice that our second exposure number of antibodies is so much faster. So we've got this slope, I have this slope, so it's a more rapid increase in the number of antibodies. The response is way faster and also much larger, which means that my body is going to kill off that pathogen before I even feel sick. We are doing this all the time. So your immune system is constantly fighting off infections that you don't even know that you have, because it's not really an infection yet, it's really just an exposure. So amazing, amazing. These memory B cells are going to confer to us long-term immunity. It's really hard for us to get the same disease more than once because we have these memory B cells. One of the ways that we can kind of trick our body into doing this so that we can do this is through inoculations or vaccinations. So immunity is based on this idea of recognition of self and destruction of the foreign material. These vaccinations are purposeful exposures to stuff that's not ourselves, purposeful exposure to foreign materials, and that's going to help our bodies learn what these antigens look like. But the vaccinations are not going to be the full-blown germ. We're going to take just a tiny little piece, a fragment of the germ or the pathogen. We might have weakened or dead pathogens that we are purposefully giving ourselves, and then our body will still respond to that because those antigens will be recognized, and then our bodies will make helper T cells that will activate the B cells that will become plasma cells that will make the antibodies and the memory cells. These antibodies and memory cells are going to hang around, and so maybe this is my chickenpox vaccine. I make some antibodies, some memory B cells against the chickenpox viral particles that were introduced to my body. But then if I ever am exposed to the real chickenpox virus, very rapidly my body can get rid of it before I even feel sick. So I don't even know that I might have chickenpox because my body has kicked it out so fast. It's amazing. More recent technology, so rather than using pieces of pathogens or dead pathogens, weakened pathogens, we now have these mRNA vaccinations. How these work, so we take a little bit of messenger RNA. For COVID, this messenger RNA is actually the code to make one of the spike proteins that is on the viral coat of COVID-19. So we take that messenger RNA, we put it into a little bubble of lipids, and then we put that bubble of lipids into our bodies. Because it's made of lipids and our cell membranes are made of lipids, there's going to be a lovely little bit of endocytosis. The messenger RNA that we made in a technology, in lab with a technology, is going to be inserted into our cells. Our ribosomes are going to take that messenger RNA and we are going to make those spike proteins. We're not making the whole virus, we're only making this one protein thing. We're going to send some of those proteins to our cell membranes. These guys are now antigen presenting cells, and our immune system is going to be like, oh, hey, that's bad. Let me come and attack this. And so our cell might actually get gobbled up a little bit by a macrophage, but in taking out this one or hundreds, not that many, we have millions and millions and millions, so losing a couple hundred is not a big deal. We drop about 20,000 skin cells every day, so it's not a problem if we kill a couple of our own cells. Anyhow, so the macrophages are going to come and gobble up our cells that have these viral spike proteins on them, and learn how to kill off the virus. We might also use some of our major histocompatibility complexes, and I'll talk about that in a couple more slides. And these guys might also go ahead and present some of that antigen, and then we're going to have our helper T cell is going to come and get activated. That helper T cell will activate, once it is activated, it'll activate some B cells, and then we'll have the plasma cells, and we'll have the memory B cells, and then we are immune. We have a defense against COVID-19, which is amazing. So vaccines actually started with this guy, Edward Jenner, amazing and terrible all at the same time. So Edward Jenner, the story goes that he took an 8-year-old boy, perhaps his gardener's son, and purposefully gave him cowpox. Cowpox is a virus that is pretty common, especially in milkmaids. So girls whose job was to milk cows would often get cowpox. And then Jenner noticed that the milkmaids who had cowpox were not dying of smallpox. So Jenner here took this child, and gave him cowpox on purpose. And then he exposed the boy to smallpox to see if the boy would get smallpox, which is deadly. Discovered that the boy was not getting sick of smallpox. And the reason is this virus that causes smallpox is very similar in shape to smallpox. And so what was happening was that the boy was making antibodies against cowpox, but the shape was close enough to also bind to and to knock out the smallpox. So once he noticed that the boy survived his exposure to smallpox, he gave himself and 23 other people, mostly in his family, cowpox, because the boy survived. Good great, amazing medical advance. Terrible human. Why? Why an 8-year-old boy? I don't even know. Anyhow, the reason that we call vaccines vaccines is because vaca, there's only, I think, one C in Spanish. Vaca is cow. Cowpox. Vaccines, vaca, cows, cool beans, right? All right, let's talk a little bit more about some of this MHC protein major histocompatibility complexes. So we have these big chunks of proteins that are on all of our nucleated cells, major histocompatibility classes, that allow our immune systems to know that our cells are our cells, and if you have a different MHC, then you're not our cells, and we're going to attack you. Interestingly, our red blood cells do not have nuclei, and so they do not have these MHC classes. There's no major histocompatibility protein complexes on our red blood cells. That makes it a little bit easier for us to give blood transfusions than it is for us to do like full-on organ donation transplants. Our red blood cells do not have those major histocompatibility complexes, but they do have these glycoproteins that give us A, B, and O blood types. We did this back in genetics, so I'm going to go super fast here, but basically if you have type A blood, then you make these type A glycoproteins on your red blood cell membranes. If you're type B, you have the type B. AB, you're going to have both B's and A's, and if you're type O, you don't have any of them, no A's and no B's. So that means that your body thinks that A's and B's are not you, and so you're going to make antibodies against type A and type B blood because it won't look like you. And so if a person with type O blood is given type A blood, their antibodies will attack that type A, and it will be bad. It'll be bad kind of like this. So let's say that this is that type A blood, and we gave it to someone with type O blood. That person with type O blood is going to have anti-A antibodies. So these antibodies are going to bind to the glycoproteins on the type A blood cells, clump them all up, and then break down those red blood cells. The problem is when we have this clump happening all over the whole entire body, all the time, even in the capillaries, super bad, lots of tissue damage. And then also all those red blood cells that we gave to that person because they needed blood are now dead red blood cells, but they may have killed the person already, so it might be too late. So super important to make sure that we are typing and cross-matching all of our blood before we give anybody any transfusions. So how do we know what our blood types are? We actually make antibodies on purpose. So we in the laboratory. So we have these things called monoclonal antibodies that we make in labs. So they are artificially derived from a single B cell clone, which is why we call them monoclonal. We have one cell and we're making lots of copies of it. So what we do is we take some type A blood, and I put that into a mouse. That mouse is then going to start to make plasma cells, helper T cells, B cells, and then plasma cells that make antibodies that are specific to fight type A antigens. We then take those plasma cells out of the mouse. We actually fuse the nucleus of the plasma cells with some tumor cells. These tumor cells, like literally cancer tumor cells, go through crazy fast mitosis. One of the things that's bad about tumor cells is how fast they divide. But in the lab, if I want a lot of cells really fast, I should use tumor cells. So we take the nucleus out of a plasma cell, we fuse it with these tumor cells. It's going to go through crazy fast mitosis because of those tumor cells, and we call these cells hybridoma cells. They divide rapidly, and all of them are clones. They are monoclonal cells. Hybridoma cells are clones of these plasma cells that are all going to make antibodies against type A blood. All of these plasma cells, these hybridoma cells, are going to start pumping out huge quantities of these antibodies that are specific to whatever antigen it was that we exposed our mouse or rabbit or whatever to. Some uses of monoclonal antibodies: blood typing, like I was explaining on that previous slide. We can also treat some diseases like rabies and cancer with these antibodies. So we can make antibodies that are specific to rabies. You give the antibodies to the person. They don't last forever, so it's not like a long-term immunity. It's not a vaccine against rabies, but it's a good treatment. And so then The antibodies will bind to the rabies particles and then prevent them from getting the person sick. We can also use monoclonal antibodies to detect this protein called human chorionic gonadotropin. And what that means is we can make pregnancy tests with these monoclonal antibodies. So human chorionic gonadotropin is a protein that is produced by an embryo and then is released in a pregnant woman's urine. So that protein, human chorionic gonadotropin, released in the urine if the woman pees on this stick, and she does have that protein in there. That protein will bind to the HCG in the urine, and then we have a lovely color change that lets the subject know if there's an embryo or not. So monoclonal antibodies allow us to make pregnancy tests. Crazy cool, right? All right, we're going to flip over to this idea of allergens. So histamine is our protein that causes these allergic responses. What's kind of weird about allergens, we think we're sick. We think there's a pathogen, but there's not really. But we think there is, and so we have this huge immune response that's a little overdone and definitely not necessary. But the pathogen might not be harmful, but sometimes the reaction is. Most of the time, allergic responses are localized. You get a bee sting, you swell up where that bee stung you, but sometimes we have major anaphylaxis where we have a huge systemic allergic reaction. Our lungs start to swell closed. We can't breathe. This is super, super bad. If you have any severe allergies, hopefully you have EpiPens to help counteract these histamines. So histamine, this is all it is, like such a tiny, tiny little molecule to create such mass hysteria in our bodies. But these histamines released by these mast cells, which are a special kind of white blood cell, is just going to lead to all those symptoms of allergic reactions. So most of the time we have to be sensitized to something, like we have to have that initial exposure to something that's going to make us allergic before we can be allergic to it. Then that secondary response is when we have so many of those histamines being released, and that histamine is what's going to lead to all of those symptoms that we feel when we have an allergic reaction to something. Inflammation is one of those symptoms. Usually it's a really good thing because it's going to draw more immune cells to a region where we have an infection. But of course, if we don't need to have that immune response, then this is not a comfortable thing. So that inflammation can lead to our blood vessels widening a little bit. That just helps the white blood cells get to the area of infection more easily. We're also going to make the capillaries more permeable so that those white blood cells can ooze out of the capillaries and into the body tissue. But then other fluids are also going to leak into that area, and that leads to the swelling. Good. Interestingly, some pathogens are species specific and some can cross barriers, like the bird flu started in birds and then it moved to pigs and then it moved to humans. And so sometimes a pathogen will infect only one particular species. Like there are some things that only make us sick, don't make anybody else sick. But then also they can sometimes cross over different species. Let's talk about some antibiotics. Amazing, amazing discovery by Fleming. So antibiotics are produced mostly by fungi, and they're going to block processes that occur in prokaryotes. So they only target prokaryotes. And what they're going to do is either stop metabolic enzymes in bacteria. They might stop cell wall synthesis, so that's what penicillin does. It actually prevents bacteria from being able to make new cell walls. If a bacteria cell does not have a cell wall, it cannot really survive very well. Some antibiotics will shut down DNA synthesis in our prokaryotes. Some of them will break down cell membranes. Some of them inhibit ribosomal action. So all of these are going to block the metabolic processes that are in bacteria in prokaryotes. So only of prokaryotes. So antibiotics will not inhibit our enzymes because we are eukaryotes, not prokaryotes. Tragically, antibiotics will also not inhibit the metabolic enzymes of viruses because viruses don't have metabolic enzymes. They don't have much of anything. And so antibiotics are entirely not effective against viruses because viruses don't have any of these things that bacteria have that the antibiotics can act against. So quick look at bacteria versus virus. Bacteria it's not a super fancy cell, but it does have cell wall, cell membrane, some cytoplasm, that nucleoid region where we have that circular DNA. It's got some ribosomes. It's going to carry out metabolism. It has proteins, it has enzymes, it does stuff. That's very different from the virus. So the virus usually comes just with a protein coat or some other kind of capsid protective coating around the outside, and then it's going to have RNA or DNA depending on what kind of virus it is. So no enzymes, no ribosomes, no cell wall, nothing that those antibiotics are going to infect, not infect, affect, inhibit. Let's talk about some viral life cycles, and I put life cycles into quotes because viruses aren't actually living. We don't consider them living because they do not have a metabolism, which sadly is why antibiotics don't work against them. So two different kinds of cycles in the viral not living cycles. So the lytic cycle, this looks really bad. So here we have a virus. This is actually a bacteriophage. So this is a kind of virus that actually infects bacteria, which is pretty cool. So the bacteriophage here is infecting the bacteria. It's inserting its genetic material into the bacteria. The bacteria cell's metabolism is then going to be hijacked and used to make lots of virus babies. The virus babies get put together in the cell. The bacteria cell explodes and all the virus babies are released, and that looks bad. The lysogenic cycle looks like it's not that bad, right? Because here's my bacteriophage. It is infecting the bacteria cell. It's putting its genetic material, inserting it into the DNA of the bacteria cell. And that's all. It stops. And that bacteria cell goes and makes babies, and we have more binary fission and more binary fission. And something happens at some point that activates the virus. And then all of a sudden, the virus is going to break into the lytic cycle, where we make lots of virus parts, and those viruses will explode from cells. This is way worse, even though this first part of the lysogenic cycle is like, well, that's not so bad. So what if we've got some viral DNA in our DNA? Problem is, all of these cells that are descendants of this cell are all going to be full of viruses. And so we have this huge number of viruses that are being released if those cells are all triggered at the same time. So lysogenic is kind of worse, just because it rolls into the lytic cycle. All right, so let's talk about HIV. So all of those words about viruses would lead us to HIV. And this is our last topic of the day, my friends. We've got just like 2 more slides. We're almost done. So HIV stands for human immunodeficiency virus. It is a virus, so antibiotics do not work against it because it does not have its own metabolism. Immunodeficiency, so it is going to cause some kind of deficiency in our immune system. So viruses, like lots of other things, are specific. So they have a specific shape, they have specific proteins on those protein coats, and they're going to bind to specific kinds of host cells. And what's really bad about HIV is that its host cell is helper T cells. So we've got our HIV virus. It is going to bind to a helper T cell. It's going to send its nucleic material into the cell. The host cell is going to make new virus babies, and it's going to, at some point, we're going to have lysis of these helper T cells. Helper T cells. Think about the job of the helper T cell. We have our macrophage that's going to eat up some pathogen. It's going to become an antigen presenting cell. It's going to activate our helper T cell, which is then going to activate our B cells, which is then going to differentiate into plasma cells, which is then going to make so many antibodies. But if I have a virus that is killing off my helper T cells, guess what? None of these things happen. So the problem with HIV is that it literally kills off our immune system because it knocks out those helper T cells. If I don't have helper T cells to activate the B cells to turn into the plasma cells to make the antibodies, then I don't have much of an immune system at all. And that's why HIV is so bad. So HIV can sometimes become AIDS. AIDS stands for acquired immunodeficiency syndrome. So if that HIV, if that virus has knocked out These are our helper T cell numbers. If our helper T cell numbers are way, way down because the virus is way, way up, then we're going to get sick from infections that really should not make us sick. So little things, diseases that usually only infect chickens now are going to kill us. So this is the problem with HIV, why it is such a debilitating disease, is that it literally inactivates the majority of our immune system. No immune system to combat any diseases at all. All right. Last idea, and then we are done, my friends. So just a quick check on transmission of HIV. I know that you know this already, but just to make sure. So how do we spread HIV? It's all about exchange of body fluids. So sexual contact is probably the most prevalent, common way still today, but pregnancy, childbirth, and breastfeeding can pass the virus from mom to offspring. IV drug use, dirty needles, it is super bad. Occupational exposure, so people who work in healthcare who might be exposed, needle sticks on accident is bad. And then also blood transfusions, organ transplants coming from people who have HIV is another way to get it. And on that not most joyful note, at least we're done. So objectives accomplished. We defined lots of vocabulary: pathogens, disease-causing agents, antigens, the pieces of the pathogens that actually trigger an immune response, leukocytes, lots of white blood cells like macrophages that will engulf pathogens. Lymphocytes are B cells and T cells. We talked about that nonspecific or innate immunity. Those macrophages will gobble up anything that doesn't belong in the body. We talked about the clotting cascade, how we have those clotting factors and platelets that will convert prothrombin into thrombin. Thrombin will then convert fibrinogen into fibrin and make a scab. We talked about activation of that specific or adaptive immunity. So we have those helper T cells that will activate the B cells that will become plasma cells that will make antibodies, but we can also have those memory B cells, which is that secondary immune response after the infection or vaccination. We talked about antibodies, and we're not going to panic because we know that this is our mnemonic device. Remember that antibodies can precipitate out pathogens, agglutinate pathogens, neutralize toxins coming from pathogens, stimulate the inflammatory response to get more immune cells to where the infection is, and they can also, we have that complement protein complex that will drill holes into bacteria so that they die. Good. We describe the production and uses of monoclonal antibodies, so we can take a plasma cell, combine it with a tumor cell. We're going to make that hybridoma cell that can make antibodies specific to something, and then we can use that for blood typing or for rabies treatment or for pregnancy tests, which is super cool. We talked about allergic responses, all about that histamine. We talked about how antibiotics treat some diseases, bacteria, but not others, not viruses, and not diseases caused by eukaryotes, and also tragically not diseases caused by antibiotic-resistant bacteria. But there are still some bacteria that we can kill off with antibiotics. Last but not least, we talked about the effects of HIV on the immune system, and the reason that it has such a huge impact on the immune system is because it attacks helper T cells. You did it. Good work.